Hello and welcome to our today's webinar, Characterization of Non-Compendial Reference Standards for Impurities, How Good is Good Enough. My name is Christian Seiner. I am the product manager of LGC, and I am quite pleased today that we have Dr. Bernard Olsen with us presenting this webinar on behalf of LGC today. And I will introduce him in a few seconds. I would like to give you a few words about Micromole, which is our brand for pharmaceutical reference standards. And under that brand, we have over 5,000 impurity, API and excipients reference standards for more than 1,000 API families. They are suitable for qualitative and quantitative applications in method development, in validation, and in routine quality control. They come with an extensive certificate of analysis. There's one for the impurity standards, and we also have certificates of analysis, more extended ones for API reference standards that are produced under the ISO Guide 17 or 34. We can also give expert technical support should you have questions to the reference standards. You will see that maybe afterwards when we are dealing with the questions. And we can also give advice on technical applications for pharmacopoeia methods or also for internally developed protocols. Right. So with that, I would like to give over to Bernard Olsen, which I quickly would introduce now. Bernard is a chemical doctor, PhD, and he received his undergraduate degree from the Nebraska Wesleyan University. And the doctorate he did in analytical chemistry at the University of Wisconsin-Madison. He is an independent consultant providing expertise in chemistry, in manufacturing, and control to the pharmaceutical industry. He has over 37 years of experience in drug development, including 29 years at Eli Lilly, where he was a senior research fellow and where he contributed to the development and support of over 25 commercial drugs and numerous developmental drugs. He has published and given invited lectures on a wide array of drug development and analytical topics, including high-performance liquid chromatography, method development and validation, impurity determination and control, genotoxic impurities, physical properties characterization, drug counterfeiting, regulatory aspect of drug development, and also quality control. And he's also a fellow of the AAPS, the American Association of Pharmaceutical Scientists. And with that, Bernie, thank you and welcome for being here today, and over to you.
Okay, thank you very much, Christian, for that introduction. I've worked with impurities pretty much my entire career, so it's a pleasure to be here today to talk about characterization of reference standards. Just a couple of scope statements to start with. Just to emphasize, we're talking about non-compendial reference standards for impurities today, and the scope will be synthetic organic compounds as opposed to biologicals. Also, just to be clear, I don't represent USP or any USP policies or procedures in my talk today. So when we think about impurity reference standards, in contrast to reference standards for APIs, there's really not too much guidance from regulatory authorities about characterization. In ICH Q3A and Q3B, it stated that if you use reference standards in analytical procedures for impurities, they should be evaluated and characterized according to their intended uses. And you'll hear this theme of intended use throughout the discussion today. Also, a similar document from even further back from the German regulatory authority that pretty much says the same thing. If you're going to use impurity standards, their identity must be ensured and the purity and assay should be defined. So speaking of intended uses, the intended use determines how much the extent of analytical effort that needs to go into the characterization of those standards. And there are two major types of uses. One, qualitative use, where we may use an impurity reference standard in for example, a system suitability test, where we're using it to check the separation perhaps between it and the API or it and another impurity to show that the method is performing as expected. Another useful qualitative use is peak identification, and in my opinion, this is especially important when you have the potential for several impurities that appear in the chromatogram close together. It's easier to use relative retention times when the impurities are far apart. But if they're close together, relative retention times can vary and sometimes not be as reliable, certainly as having a standard or a peak marker. Using impurity standards in a qualitative way for validation of specificity, and in fact, I would say even in development of separations is another good qualitative use. Of course, the other major type of use is quantitative, and we can have the case where there's a limit test, where we're trying to determine whether the impurity is present above or below a certain value. Or we can have often cases where we want to quantify the impurity and return a numerical result for it. The quantitative use also extends to validation of accuracy for quantitative use. So first, let's address qualitative use. What should be done to characterize standards for that purpose? Well, the identity must be known for qualitative use. You want to make sure you have the correct compound. And to do this, a combination of techniques is recommended. Proton NMR is very powerful to establish consistency with the structure that is proposed for the impurity. The infrared spectrum should also be consistent with the structure. And also doing carbon, hydrogen, and nitrogen analysis. And we'll show several examples where this can be important, especially when we're trying to distinguish free acid or base forms versus salt forms. The molecular weight should be consistent with the structure, and this is usually obtained from mass spec coupled with LC or GC. And we want the UV vis spectrum to be what would be expected for the impurity structure at hand. Also, even for qualitative use, we should have some expectation of the reference standard's purity. This is usually estimated from an LC or GC impurity profile. Sometimes quantitative NMR can be done for this, but a good target is to have greater than 85% purity. If you're lower than that, sometimes that can create difficulties in interpretation of the NMR or IR spectra because of the presence of other components. So 85% is a good target to have, even for qualitative use. So for quantitative use, the practical accepted approach, of course, includes identity. Again, we want to make sure we have the correct compound in hand, and the same techniques would apply as for qualitative use. Here, knowing whether you have in the case of ionizable compounds, the free base or acid versus a salt form is even more important for quantitative use. And again, the other spectral properties should support the identity. However, for quantitative use, we also need to have an assay value assigned in order to use in the calculation for quantifying an impurity. This is often determined using the so-called 100% minus method, sometimes referred to as the mass balance method or approach. And the calculation is shown over here in this formula. Basically, it's taking 100% minus other components present in the material. Usually water is considered, done by Carl Fisher with coulometric mode, usually takes less sample. Residual solvents that might be present can be determined many times by proton NMR, or if necessary, GC with headspace sampling can be performed. Those percentages are subtracted from 100%, and then the result is multiplied by the relative chromatographic purity percentage to get the assigned assay value. And if available and sufficiently specific, another technique such as quantitative NMR, which is becoming more popular these days, can also be used to assign percent assay. LGC indicates that the certificates of analysis that they supply have been regularly accepted as for impurity standards by regulatory bodies. And here's an example for this compound, showing some basic information on the first page, including the storage conditions and the assigned assay value. Along with that, the identity information is provided, so the proton NMR spectrum that is consistent with the structure, also the mass spectrum value, and the IR spectrum, all consistent with the structure of the material. Then information about the purity is given. Here the HPLC conditions used to look for potential impurities, and the chromatogram, in this case showing a small impurity at 0.03% before the main peak. Along with that, the water content, in this case 0.22%. And then finally, residual solvents, which were quite low for this material. And then the calculated the 100% minus method was used, and the results shown here as 99.6. So you know the basis for determining that result. Now let's go a little deeper into some examples of other points to consider during characterization. And I had mentioned that often carbon, hydrogen, nitrogen analysis, which is sometimes underestimated tool, can be quite useful. And especially when we have the potential for free forms, acid-base, versus a salt of those forms. This is especially important if the reference standard is going to be used for quantitative purposes. And it can be an issue when switching from one supply of reference standard to another, and we'll show an example of that. But just to illustrate, here's for this impurity standard, if we have the free base, the theoretical amounts would be shown in this this middle row, and for the hydrochloride salt, those results are, as expected, lower because of the presence of the counterion. In this case, the result for the material corresponded with the theory for the free base, so that helps confirm that that's indeed what was present. So in this case, for the chlorophenylpiperazine hydrochloride compound shown here, we have several possibilities. From the literature, we could have the free base, which is actually a liquid with this molecular mass. The hydrochloride salt, as illustrated, has, of course, a higher molecular mass and a melting point of 210 to 214. And the dihydrochloride has the that can be potential, has a even higher mass, and actually the melting point is a little lower. Well, this material is available from several commercial sources, and when we look at these in the catalogs, we see supplier 1 listed a molecular mass of corresponding to the free base, the CAS number corresponding to the hydrochloride salt, as well as the molecular melting point corresponding to the hydrochloride salt. But in fact, when purchased, that material was the dihydrochloride. Another example listed a molecular mass corresponding to hydrochloride and the CAS number and melting point, but as before, when purchased, the dihydrochloride was actually what was supplied. Finally, another supplier listed the hydrochloride mass, this time with the dihydrochloride CAS number and dihydrochloride melting point, but in fact, the hydrochloride salt was what was furnished. So this shows some of the variability that can be obtained depending on the source of the material and the care and characterization that is necessary to know what indeed the standard corresponds to. Another point to consider also relates to CH&N analysis, and this is to consider when water or residual solvents might be present. And the example in this case is risperidone cis N oxide, where in this case the theoretical values given in the middle row here were higher than the obtained values, which suggests that something else is going on. And in fact, this is a case where the amount of water needed to be considered. This had appreciable level of water, almost 6.5%. And if one corrects the values obtained for the amount of water that was present, actually you come up to be very close to the theoretical for the compound expected. So that again shows how CHN analysis can be useful. Another theme here in characterization is consistency among the results obtained. And the first example here is looking at proton NMR and HPLC for 4-chlorobenzoic acid, where the HPLC purity was 99.3%. And in the chromatogram, we see that the main impurity was this peak in the front here. Well, at that level, you should expect to see something in the NMR. And in fact, you do see resonances corresponding to the aromatic protons for whatever this impurity happened to be. So those two techniques are consistent and support the results obtained. Another example where you're trying to check consistency was this compound, where in the bumetone we see the HPLC gave a significant impurity for this reference standard at about 5.9 minutes. This is almost on the order of 10% of the main peak or so. And again, you would expect to see something corresponding to that in the NMR. However, we look at the NMR and we see here shown in red just a small signal for a little bit of residual dichloromethane. These signals correspond to the DMSO solvent that was used. And of course, we have the trimethylsilane marker, but nothing else really showing up in the NMR that would suggest there's a 10% impurity present in this material. So what's going on? Well, here we have to remember that other things can happen. In this case, it turns out that this compound was light sensitive. And in fact, we could induce isomerization where the trans isomer on exposure to light in the analytical solution forms the cis isomer, and actually a freshly injected solution shows a single peak, whereas the cis isomer grows in with time. So that's important to consider what might be happening during analysis as well. One point that comes up often is, can I use a qualitative impurity standard, such as obtained in the research lab, for quantitative applications? And we'll see that the answer is yes, that's possible, but you need to be thinking about your intended use. And for example, sometimes an impurity is given as present at greater than 80% purity or something similar. So if you're going to use such a standard as a quantitative standard in calculation, what purity value should be taken? And I think we're going to pause and take a quick survey to see what the attendees think. In this case, if it's labeled greater than 80%, should I use 80%, 90%, 100%, or really don't know what should be done there?
Okay. Yes, thank you, Bernie. And Bernie mentioned already, there's a short survey here, one question that we have for you. where you can give your vote or your answer. If the purity of the impurity standard is indicated, for example, with more than 80%, or in a similar way, with what value should I calculate? And Bernie asked already, should you calculate with 80, with 90, with 100%, or do you have no idea? So yeah, please, if you could give your votes, that would be great. So and afterwards we are showing then the results of this this quick survey. So I see at the moment that 30% of you have voted. Let's see that we get minimum 60%. That would be great. 70% would be even better. We are now at 50%. So please, could the others give some votes as well? We are at 56% now, so we would need a few more clicks. And well, if you don't know what to use, you can also click on I have no idea. We will in the end explain what is the right value that you should use here for this kind of example, for this impurity standard. So we have now 60% and 63. We are coming close to 70, and then we can stop the survey and see the results. I can play back the results then. So right, 65%. Anybody who wants to vote here still? I think it's not the case. So let's we are at 67% participation. The result is quite interesting. So I close now the survey. The result is quite interesting, and I will show the result now here. Right. So we have roughly 2/3 that's saying we should, of those that answered, we have roughly 2/3 who said I should calculate with 80%. 3% said you should calculate with 90 because it's in the middle of 80 and 100, and 8% said you should calculate with 100%, and nearly more than a fifth of you said you have no idea. Okay, so perfect. Thank you very much for having the survey, and yeah, I will take it out now, and over to you, Bernie, again for the right answer, please.
Okay. Well, this is always very interesting to illustrates kind of the confusion that can come about with a question like this. And basically, the answer is to not calculate with the minimum value of 80%. And why would we not want to do that? The thought is that the material may be and is probably purer than that value, and you risk underestimating the impurity in the sample if you use a value of 80%. And if at all, it would be better to calculate with 100%, because there the risk is that you would overestimate the amount of impurity in the sample. And you know, why would we want to overestimate and have that risk? Well, the main thing is because it errs on the side of safety. You would fail a specification or exceed a threshold level, and it would not be a case where the impurity would really be out of spec, and by underestimating you would have it within spec or within a threshold. So that's the main reason to be conservative and use 100% for that situation. However, we know that that brings an economic risk for a pharmaceutical manufacturer, that if you're overestimating, you could have a false positive out of specification result that would lead to unnecessary investigations. If you're in development, you might exceed a qualification threshold for an impurity and then need to start considering qualification studies such as animal tox type studies. So some more on this overestimation risk. And basically here, if you're using a qualitative standard, the idea is that the fewer analytical details, the less information you have, the higher the economic risk you take on. And as we've seen previously, depending on the sources, qualitative standards may not have the correct identity, especially in regard to salt forms. Residual solvents and water may not be checked in some of these materials. And as we know, in the case of solvates or hydrates, these solvents can be an appreciable percentage of the material. So, show an example of that. Let me come back to this statement. Even if we have the chromatographic purity quite high for a material, if we don't know the correct salt form, we can overestimate by quite a bit. And I prepared this little graph that shows on the x-axis the true assay value of a material, and then on the y-axis the percent error when you assume that the material is 100%. So, of course, when the true value is 100%, the percent error is 0 because you've accurately assessed it. If we go to an extreme example at, say, true assay of 50%, now you would overestimate by a factor of 2, or your error would be 100%, in assuming that this true assay was 100%. And in the example shown here, where the true assay is 70%, which you can have in the case of some salts, and you assume that to be 100%, your error will be over 40%. And an example where that can be important is shown here, where you have in actuality a 0.12% impurity. If you assume and overestimate the standard impurity by assuming 100%, you would calculate 0.17%, and now you've exceeded the 0.15% qualification threshold, and may be necessary to consider tox studies to qualify that. So that's the economic risk. Another question that comes up sometimes is a second assay method needed for quantitative impurity standards, as is the case for a primary API reference standard? And LGC gets these requests sometimes from clients who in turn are getting these requests from some regulatory authorities. So just for my personal opinion, that for impurity reference standard, this really doesn't add a lot of value if in the case where assay has been assigned using the 100% minus method or quantitative NMR, that the risk of underestimating the assay of the impurity standard is very low. Also, well, and that means that there's a low chance to underestimate the impurities present in the API and so the safety risk for patients is low. So it really doesn't add value from a regulatory standpoint to need a second method. Also, the overestimation is minimized when the appropriate identification and assigned value have been carried out. So we talked about several examples of characterization of impurity reference standards. And again, I would like to thank LGC for the opportunity to discuss this today. Also to these LGC scientists who furnished the information in the case study slides. And of course, to you, the audience, for your attention. We, I think, have the opportunity for questions to be forwarded in the question box. And as Christian indicated at the start, some of these he can take by email as well. And we can perhaps address some questions now. And if not, I'm sure LGC will be following up to respond to others received. So thank you again, and Christian, back to you.
Yeah, thank you, Bernie. Also from my side, as Bernie said, there is now time for questions. You can also send the questions later to me. Thank you very much. If there are any further questions, please let me know by email. And apart from that, I wish you here in Europe a good afternoon, or over to the States or the Americas. Should there be anybody from the Americas listening, a good morning again. Okay, thank you very much. Bernie, any final remarks from your side?
No, thanks for the opportunity to discuss this important topic.
Okay, so thank you very much. And yeah, let's hope we will meet again online on one of our next webinars, which we will make available to you and let you know in due time, of course. Thank you, and bye-bye.